Zoonotic diseases pose a significant threat to human life, which is exemplified by the recent deaths caused through cross-species infections, namely through human interaction with horses. Prevalent in bats, zoonotic diseases such as the Hendra virus are transmitted to horses through consumption of foods contaminated with the saliva or urine of an infected species. With over 30 horse and 4 human deaths recently recorded, further research into the dynamics of Hendra virus in bats has been commissioned in order to better understand the deadly disease. In November 2004, Raina K. Plowright and 7 other researchers began their research for the article, Reproductive and Nutritional Stress are Risk Factors for Hendra virus in infection in little red flying foxes. They hypothesized that pregnancy increases the little red flying fox's susceptibility to the disease, as well as hypothesizing that the seasonal increase in contact rates via mating, as well as the effects testosterone associated with mating has on the immune system, may drive temporary fluctuations in the Hendra virus dynamics, as well as risking a spillover to horses and humans. Tests were conducted from November 2004 to March 2006 in the Northern Territory during spring, autumn and winter, which coincides with the mating, late pregnancy and early birthing, and late lactating and early weaning stages. To capture the little red flying foxes, mist nests were used in the fly-ins, where bats were caught and then administered an anesthetic via mask induction of isoflurane. Captured bats had the following criteria recorded for age classification. They also had blood samples collected from the cephalic vein, which is the leading vein along the edge of the wing membrane. Before being released, each bat was also fitted with a thumb band for future identification. To test for the Hendra virus, a serum neutralization test was performed on serum plasma samples from lab bats. If the Hendra virus was neutralized at a dilution of 1 to 5 or greater, it was regarded as a positive test. If the sample did not neutralize, it was regarded as a negative test. With some samples became toxic to the cell layer, which were recorded as unknown. Over the research period, 664 flying foxes were sampled, with 601 S&T results coming back positive. The 2005 birthing season data showed the seroprevalence in pregnant and lactating females was more than 4 times the seroprevalence in the adult male. These results supported the hypothesis that pregnancy increases the little red flying fox's susceptibility to the disease. Seroprevalence during mating and birthing seasons was also found to be more than twice as high as the seroprevalence in the late lactating weaning seasons. These results also were supported by the second hypothesis. In summary, they found that the pregnancy and increased contact rates, as well as the effect of testosterone during mating and mating season, increased the bat's susceptibility to the zoonotic disease. This then had an increase in risk of spillover to horses and humans. The entire article, Reproductive and Nutritional Stress are risk factors for Hendra virus infection in little red flying foxes, can be found on the Proceedings of the Royal Society site. Thank you for attending.